schools, specializing in providing supports to students in areas of executive functioning, social learning, social pragmatics, and self-regulation. Although we are not diagnosis-driven, we do serve students with high-functioning autism, social pragmatic disorder, sensory processing disorder, communication challenges, ADHD, anxiety, depression, and executive dysfunction. We have two campuses, one in Chantilly, Virginia, serving children grades K through 12, and the other campus in Silver Spring, Maryland, serving children K through 8. Please visit our website for additional information, including our open houses and the admissions process at www.theauburnschool.org. And now it is my great pleasure to introduce Dr. Bruce Kerr, founder of Potomac Psychiatry and a great partner in this series. Dr. Kerr?
Thank you, Andy, for that wonderful introduction and for the opportunity to present this series along with the Auburn School. Good evening, everyone, and welcome. My name is Dr. Bruce Kerr, and I'm a psychiatrist and the founder of Potomac Psychiatry, and thank you for joining us. At Potomac Psychiatry, we don't just treat our patients' symptoms, we uncover and treat the underlying root causes of their mental health conditions using recent advances in genetic testing, gut microbiome assessment, which we'll hear about this evening, cellular health and immune system functioning, and other cutting-edge laboratory tests. We practice what's called root cause psychiatry. This is the third session of our four-part virtual webinar series, where top experts in autism will be available to answer your questions. Potomac Psychiatry, in concert with the Auburn School, has pre-recorded 6 highly informative video webinars from these experts. And at the end of this session, I will let you know how you can view these webinars online and be able to attend the upcoming Q&A events. And now it is my honor to introduce a true superstar in the field of science, Dr. Rob Knight, a world-renowned researcher in the field of gut microbiome science, including the role of the human microbiome in autism. Dr. Knight is kind enough to be joining us from South Korea, where he is presenting at a global conference and where it is also 7:30 in the morning. Dr. Knight has a very distinguished career, founding director of the Center for Microbiome Innovation and professor of pediatrics, bioengineering, and computer science and engineering at University of California, San Diego. He is the Wolf Family Endowed Chair in Microbiome Research at Rady Children's. He is also a fellow of the American Association for the Advancement of Science and of the American Academy of Microbiology. And he received a distinguished honor. In 2019, he won the Director's Pioneer Award for his microbiome research, along with the Massey Prize, often considered a predictor of later winning the Nobel Prize. He's also the author of 2 books, Follow Your Gut, The Enormous Impact of Tiny Microbes, and Dirt is Good, The Advantage of Germs for Your Child's Developing Immune System, and has written over 850 scientific articles. He has spoken at TED. His recording there has been viewed over 2.1 million times. His laboratory has produced a lot of software used in research throughout the world, and his work has linked our gut microbiome to a range of health conditions, including autism, obesity, and inflammatory bowel disease, which has enhanced our understanding of microbes in multiple different environments. And he's worked with thousands of researchers around the world to create new knowledge. To ask Rob a question this evening, at the bottom of your screen, please left-click on the Q&A button and type in your question. He may not get to answer all your questions this evening, but we'll try to get back to you with answers in the coming weeks. Once again, thank you for joining us, and please ask away.
Well, all right. Well, Dr. Knight, if you have anything that you'd like to start us off with and talk about, more than welcome, but I have a bunch of questions that are starting to come in. So up to you.
All right. Thanks, Dominic, and thanks, Bruce, for that very kind introduction. If questions are going in, let's go straight to them.
That sounds like a plan, Dr. Knight. So the first question of the night comes in. Oh, this is a good one. So Dr. Knight, oh, they watched your video. Dr. Knight, in your video podcast for this event, you mentioned that from a genetic standpoint, we are 1% human and 99% microbial. And we cannot change our human genes, but can change our microbial genes and should focus on that. Please tell us how we can focus on changing the microbial genes in children with autism to improve their communication and social skills and learning ability.
Yeah, so that's a great question. This is exactly the kind of thing where there's a lot of research going on at the moment, but not yet a lot in the way of clinical recommendations. And so what I'm going to tell you comes from several different strands of research. One is that we know that response to nutrition is highly personalized, and a lot of that personalization comes from our microbial genes rather than from our human genes. So there was an amazing research paper in Cell from Eran Segal's group at the Weizmann Institute, where what they were able to do was they were able to predict from the microbiome which food items were specifically good for blood glucose response and which were bad for individual people. And what they found when they looked at 800 people, when they averaged the results for all of the people, they got exactly the glycemic index values that you'd get if you looked it up if you looked it up on the web. But for individual people, it was all over the map. So for example, for some people, it was a lot better for them to eat ice cream than it was to eat rice in terms of their blood glucose, which is just fascinating, right? You'd never expect that. And so for blood glucose control, they were able to do things like design very specific diets, telling one person they should eat regular potatoes, a second person they should eat sweet potatoes, totally opposite consequences for their blood glucose. So we have this proof of principle that we can control blood glucose, and we can do it in a very personalized way based on the microbiome. Also in the area of cancer, Dr. Jennifer Wago at MD Anderson Cancer Center has shown that you can tell whether people are going to respond to checkpoint inhibitor therapy for melanoma based on their microbiome, and even change them from a responder category, change them from people who are non-responders, who the drug is not going to work for, into responders by modifying their diet with fiber supplements primarily. So we have this proof of principle that the microbiome controls responses to diet. We have this proof of principle that you can use diet to change the microbiome in ways that matter for those responses. Do we have that in autism? Unfortunately, not yet. So the research from other areas of medicine strongly suggests that it will be possible to do this one day for autism, but that the recommendations are going to be very different from one person to another. And we really need to do the research in children where the microbiomes are different from adults. And we really need to do it in autism populations specifically, where the microbiome is different from neurotypical populations, to get that understanding so that we can give personalized and precise recommendations. I know that's not very satisfying, but a lot of what you're seeing in the video is the results of early stage research in mice and research on other diseases. And it just takes a while to translate it to the specific condition that you're interested in.
But while we're waiting for that research, which will be profound in its influences to help children with autism, what we know is children with autism have much higher rates of brain inflammation. And for example, cerebral spinal fluid samples show 50 times the level of an inflammation biomarker, TNF alpha. Are there some basic principles parents could use around more of an anti-inflammatory diet, let's say in a more general sense, while they're waiting and we're waiting for this wonderful science to come along?
Yeah, absolutely. So in terms of anti-inflammatory diet, one thing that is helpful is that the dietary recommendations for children tend to be very similar to the dietary recommendations for adults, in terms of things like the food items in the Mediterranean diet, which are generally anti-inflammatory, are likely to be helpful in kids as well. So this includes things like substituting olive oil for butter. It includes things like reducing consumption of red meat, increasing consumption of fish. Although if your kids are anything like mine, getting them to eat fish may be a bit of a challenge. But let's see, emphasis on whole grains. Besides the Mediterranean diet, there's a number of other anti-inflammatory diets out there that are based on broadly similar principles. You mentioned TNF-alpha in the brain. Anti-inflammatory diets also tend to be good for things like increasing butyrate production in the gut and reducing gut barrier dysfunction. So essentially making it harder for microbes and their products to get across the gut barrier, which is also frequently a problem in autism.
Right, that's what I think a lot of parents would know as leaky gut syndrome. So it's a way to produce a butyrate which helps prevent leaky gut syndrome or treat it.
Correct. And so fiber is particularly important for that, where that gets fermented into butyrate in the gut, and that's very important for maintaining that barrier integrity and preventing leaky gut syndrome.
Well, thank you for that answer. Thank you. I think that covers that. Thank you, Dr. Karen, Dr. Knight. We got another question that just came in from the crowd, and we may have touched on it a little bit, but it's a little more focused here. Do you believe, based on your research, that the root cause of autism is the inflammation of that results from microbiome imbalance?
I think autism is a complex disorder with a lot of different factors and probably different causes in different children. We know for sure from animal research that in mouse models you can induce autism by changing the microbiome. Induce models of autism, I should say, by modifying the microbiome. So at least in those models, the microbiome by itself can be causal, even in a normal genetic background. But that doesn't necessarily mean that it happens the same way in humans. From my own research, especially the work we've done together with Dr. Sarkis Masmanian at Caltech, I think it's likely that for at least some individuals with autism, the microbiome is primarily responsible. But understanding which individuals that's true for, again, is the focus of a lot of research at the moment, rather than something where there's an FDA-approved diagnostic test that you could just run out and get right now. So I think it's probably true, although again, there's a lot of details to work out.
Well, thank you for answering that question, Dr. Knight. We got another one coming in. People are interested here. Could you please, and real quick, everyone that's in, keep all the questions coming in. We're going to get to them. If we don't answer them tonight on the call, we will follow up and answer them in the upcoming weeks. And so the next question that's come in is, could you please explain how the human microbiome influences the development and potential symptoms of autism in children? What are some of the key mechanisms at play?
So we think the key mechanisms for how the microbiome influences autism, and again, a lot of this comes from animal models where we think the same thing is true in humans, but it will take a while to determine what is the same and what is different. Part of it's inflammatory processes. So essentially microbes in the gut can lead to systemic inflammation, and that can lead to increased inflammation in the nervous system and the brain. Another really important factor is production of small molecules. So molecules like 4-ethylphenyl sulfate, which is a molecule produced by some kinds of bacteria that we think are bad in the gut. That by itself can cause a number of symptoms that are reminiscent of autism in animal models, and it's also been found in higher amounts in the blood of human individuals with autism. So we think small molecules like that can be produced by the microbiome, circulate in the bloodstream, and have distal effects on the brain. And we also think that gut dysfunction by itself might play an important role. So, for example, leaky gut, if microbes are getting into the bloodstream, some of the antigens that they have, like LPS, lipopolysaccharide, increases inflammation directly, and is also likely to be an important part of that process.
I think that's a really important point, that so many elements of a Western diet are pro-inflammatory, particularly sugars and simple carbohydrates, as opposed to, I know one of your recommendations that I try to follow, which is tough, is 35 different plants within a 2-week period to bring that fiber in to help a healthy gut microbiome. But leaky gut syndrome is something that can be evaluated and treated. And one of our earlier experts who spoke a couple of weeks ago, Julie Wendt, does exactly that with children in an effort to personalize it. But that leads me to another question. With these small molecules that are being identified, is there some future strategy that might be used to neutralize them? Like a vaccine, say, to create antibodies to them or something like that? What do you think?
Yeah, absolutely. So the first step is the first step is finding the microbes that are producing them and what are the pathways. One thing that's really interesting is that in other settings like cardiovascular disease, a large part of the reason that red meat is bad for some individuals from a cardiovascular standpoint is that the carnitine in the meat gets metabolized by bacteria into a compound called trimethylamine N oxide, or TMAO. And so you have to have, basically you have to have the wrong kind of bacteria and the wrong diet together to produce that molecule and elevate cardiovascular risk. So one question is, even if you have the bacteria that might be causing you problems, could you keep away the precursors that they need to make those bad molecules by eating the right diet? A second question is, how do you want to target the bacteria and reshape your microbiome? And there's a whole lot of different ways to do that, ranging from extreme measures like fecal microbiota transplant, where you literally take the microbes out of the stool from someone who's healthy and give them to someone who's sick. Right now, the FDA will only let you do that for recurrent C. diff infection outside the context of a research study. But there have been some quite promising research studies doing that kind of thing for autism directly, with really remarkable results, with improved symptoms even 2 years after the transplant was done. But then less extreme than that, you can say diet is one of the most powerful things to reshape the microbiome, but it takes a long time to take effect, typically 6 months to a year. But strategies like eliminating sugar and ultra-processed foods, things like increasing the number of plants in your diet, which has a huge impact on reshaping your microbiome, and those kinds of things are very feasible strategies. And then, just like you're suggesting, if you know what kinds of bacteria are producing the things that you don't want, you could consider targeting them with a vaccine. You could consider precise antibiotics. You could consider encouraging the growth of bacteria that compete against them, so that they can't grow and can't produce the molecule that you don't want. There's a lot of different strategies that are on the horizon there. And especially the mRNA vaccines that were so effective during COVID, they present this really exciting technology platform for being able to take out specific populations of bacteria that we'd really rather not have in the gut. Although again, so far this is early stage and only in animal models. Like for other mRNA vaccines, it usually takes 10 years or more for that to make it from an animal study into humans, although the timeline is a lot shorter for mRNA vaccines than previous technologies.
You know, in your video that you alluded to earlier, you did cite that study where fecal microbial transplantation was used in autistic children, and the 2-year follow-up was quite promising. Can you say a little more about that, and whether anyone is thinking of filing with the FDA to get approval for that as a therapeutic option? Let's say, particularly with very severely autistic children. What do you think?
Yeah, absolutely. So that study was conducted by a consortium of researchers in Arizona, including my good friend Rosie Kreshmelnick Brown, and Dr. Jim Adams, who was the lead clinician on that study. And so essentially the study design was to take fecal material from healthy children and then very extensively screen it to make sure that there weren't any pathogens, weren't any antibiotic resistant bacteria or other things that could cause problems, and then transplant. Essentially by giving some of this processed stool by colonoscopy to children repeatedly over the course of, I think it was 12 weeks, to reset their microbiome to the healthy state. And what they found in that study was that both during the series of transplants and also in the initial study, they looked at the end of the transplant series, but then they did a follow-up study looking at the children 2 years later. What they found was significant improvement both in GI symptoms and in cognitive symptoms, and that improvement was durable. So then the question is, what's the barrier between doing that and having fecal microbiota transplant, or FMT, having that as an FDA-approved therapy for autism? So the first issue with the study is it was a relatively small number of children, and it was open label, so everyone knew if they were getting the fecal transplant or not. And what the FDA usually requires is what's called an RCT, or randomized control trial, where what you do is you have a placebo. So in this case, you would set up a new study with a large number of children, where half of them would get the fecal transplant from someone who was unaffected by autism, and half of them would get their own stool given back to them, so that you had a comparison group. And you would need to do a much larger clinical trial, so you would need to do it with hundreds or thousands of individuals, rather than with dozens to get it on the market. So all of that is difficult and expensive. There's a lot of groups that are very interested in doing it, including a group here at UC San Diego. A lot of the barrier is regulatory, so it takes you several years to file and get approval for the investigational new drug application. So believe it or not, even though it's something that you literally poop out, the FDA does treat stool as a drug. And the reason they do treat it as a drug is it's a substance that's known to modify clinical endpoints. So for that reason, you have to you have to prove that you're manufacturing it in a responsible and reproducible way, which again is pretty difficult to do for poop, right? So you have to have a whole GMP manufacturing facility to take the raw material, as it were, and then turn that into something that's more like a drug in terms of its reproducibility, its safety screening, and all that kind of thing. So being able to get the manufactured material to FDA standards is difficult. Getting the IND is difficult. And those are the main barriers to this kind of research at the moment, at least in the United States. So, for example, in Australia and in the Netherlands and in a number of other countries, the regulations are much less tight. And so as a result, we're seeing much more rapid progress in those countries in this kind of research direction.
Thanks.
Well, we have another question that came in, and this question is an interesting question from the same person that asked the inflammation question. Does this model align with the PANS model for some other mental health issues?
Yeah, great question. So my friend and collaborator here at UC San Diego, Dr. Jack Gilbert, has done a fair amount on pandas with various collaborators. And with pandas, I think it's pretty clear that we're seeing an immune trigger rather than small molecule metabolites. But there's certainly a lot of shared interest in trying to understand which aspects of the neurological changes are reversible, which are irreversible, and also different strategies for targeting the microbiome as a whole, rather than just one organism at a time.
So, it's interesting in terms of diagnosing PANS, or in adults, ONDAL, with respect to Lyme disease. We use the Cunningham panel, and then arrange for different immune modulatory. treatments. But it raises the question of whether you believe the gut microbiome and leaky gut syndrome plays a role in autoimmune diseases more generally. And is there an autoimmune component to autism? We know that there are autoantibodies to folate receptors in 70% of children with autism. That's why we do the FRAT test and treat them with leucovorin. But what are your thoughts about this potential relationship in the gut microbiome and autoimmunity and brain inflammation?
Yeah, so that's a great question. And the gut microbiome is certainly known to be involved in quite a number of different autoimmune diseases. So, for example, rheumatoid arthritis was one of the first diseases that was linked to the gut microbiome and also the oral microbiome in very elegant work by Dr. Dan Littman and his collaborators at NYU. So then beyond that, we've done a lot of work with Sarkis Mazmanian at Caltech, who I mentioned earlier. With Sarkis, we have looked at multiple sclerosis and Parkinson's disease, as well as autism. And we're currently engaged in a study of Alzheimer's, where there is also some suggestion that there may be an autoimmune component to that. So it's quite well established that the gut microbiome is altered in a wide range of different autoimmune diseases. And then the question is, how do you establish causality? How do you tell whether the microbiome leads to the autoimmune disease, or instead does the autoimmune disease change the microbiome as a side effect at the same time that it's changing the rest of the body? And this is where those animal models are really helpful. So there's a technique that we can use where we take mice that are raised in a bubble with absolutely no microbes of their own, so germ-free mice, and then we can give them the microbes of someone with an autoimmune disease and the microbes of someone without an autoimmune disease, and see whether the mice given those microbes differ and whether they develop the disease. And so this has been a very powerful paradigm for looking at these relationships between microbes and autoimmune disease. And in a lot of different cases, including the ones that I mentioned, we've been able to recapture aspects of these diseases in mice, just like we have for autism. Now, that doesn't necessarily establish that there's an autoimmune component of autism that we're seeing and recapturing in these mice, but it's certainly suggestive, and it's certainly the kind of thing that would be quite amenable to experimentation to track down the specific parts of the immune system involved, the specific autoantibodies and so on. I don't know of anyone who's done that, who's combined that kind of profiling of the autoantibodies with the microbiome in the same study, although that's certainly a good idea.
Well, that raises a question, and Dr. Ng spoke about it in his webinar and in the Q&A that we did with Auburn around some researchers believe that these antibodies that develop in children to the folate receptors in their brain may originate from ingesting dairy, and that there may be some role for dairy in creating the autoimmune disease. Is there any thought you might have about how dairy might affect the gut microbiome, and is there potentially a link there, or is that, what do you think?
Well, that's certainly an interesting idea. I don't know about the context of autism specifically. Maybe you can tell me one prediction of that would be you would expect that cultures that consume very little dairy would have lower autism rates if that was a major cause. Is there any evidence in support of that? Maybe that cultures that don't have a tradition of consuming dairy, or where lactose intolerance is a lot higher, is there any link between that and low autism rates?
I don't know. I know this is an area of research that Iliad Neuroscience has been pursuing, and Dr. Richard Fry. But I think what we could do is we can look into that and then send out an answer to our participants this evening. We'll make a note to do that, and obviously you as well.
Great. Well, that would certainly be very interesting to know. In terms of does dairy change the microbiome? We certainly we certainly do pick up influences of dairy, and it's a little bit complicated. So my good friend and collaborator, Dr. Ruth Lay, at the Max Planck Institute in Tübingen, has done quite a lot of work looking at the microbiome and lactase consumption specifically. And so what is interesting there is that the microbes in your gut can produce lactase. And so there are people who you would expect to be genetically lactose intolerant who are able to metabolize lactose because their microbiome is able to pick up the slack, as it were. And this offers some really intriguing possibilities in terms of maybe modifying your microbiome, including by other aspects of diet, so that you would be able to metabolize lactose and be able to consume dairy. But part of the reason why we see a link between those microbes and lactose consumption, part of the reason is that if you have a microbiome that doesn't support eating lactose, then you're probably not going to eat the kinds of foods that would cause those microbes to proliferate. And so again, you have this cause-effect problem. In terms of relating any of that to autism, essentially you're getting at a very interesting basic principle with microbiome research, which is essentially you want to take people who are healthy and people who are sick for some reason, and then you want to ask, is there something that I know that changes the microbiome so that people who are sick can move back towards their healthy state? And the reason why that's difficult to do at the moment is that different studies use different methods that are not comparable to each other. And so as a result, we know about a lot of things that change the microbiome, and we know a lot of differences between different populations, including healthy versus various diseases, or including people with autism versus people who are neurotypical. But what we have a much less good handle on is, do the things that we know change the microbiome, change it in a relevant direction to those diseases? And that's a lot of what we're trying to create in my lab at the moment, a standardized resource where, for many different ways of changing the microbiome and also for many different health conditions, can you do them all with consistent methods so that you can start to relate all of those studies to each other, rather than having to treat each study in isolation as its own as its own finding, but you can't really connect it to the larger body of work that's been done on microbiomes in all these different settings. And if we can solve that problem, it'll make tremendously more efficient use of all the taxpayer dollars that have been poured into the NIH, into the EU studies on these kinds of things in Asia and so forth, by being able to reuse the results of that research and plug each new study into this large existing framework that lets you tell whether the results are relevant to your situation.
Well, that's amazing. I do want to honor everyone's time today. And so, Dr. Knight, I'd love to give you the chance to, you've said a bunch of wise words already. I'd love to give you the chance to give your closing remarks, inspire any kind of hope that you'd like to inspire, and then Dr. Kerr will wrap us up.
Sure, absolutely. Well, I think there's tremendous potential and tremendous hope in these microbiome-directed therapies for autism, whether we're talking about fecal transplant, whether we're talking about diet, whether we're talking about new vaccines or targeted antibiotics or phage therapy, or any of these things. But it is important to realize that a lot of these ideas are still potential for the future. Other than diet, these are not really things that you can safely do yourself. And even with diet, there's a lot we don't know about at this point. So we can give very general recommendations like eat your vegetables, don't eat too much sugar, and so on. But our grandparents might have given exactly the same recommendations, right? And so getting at the specific things that have a really large impact on each individual, that's where the science is going, but it's not quite there yet. So I think there is tremendous hope. One thing that didn't come up, which usually does, is probiotics, and I do want to refer back to that study by Dr. Wago at MD Anderson, where she was reshaping people's diet so that as cancer patients, they would respond to a treatment rather than failing to respond to that treatment and dying of cancer. And I mentioned how fiber interventions were extremely valuable in that population. They also looked at probiotics, and when they gave the probiotics, 2 different commercially available ones, to mouse models of their cancer patients, the mice died earlier. They also noticed that their cancer patients who were taking more probiotics died earlier as well. So a lot of the stuff is very counterintuitive. Just because something is good in what's considered a normal or healthy population does not mean that it's necessarily going to apply in populations with different conditions. So you do need to be very careful about that. But I think we're at exactly the point where it's really useful to track the research. There are microbiome-directed therapies for other conditions coming out on the market right now, like for IBD, for example. And I think together as a community, getting those therapies to where they can be useful for individuals with autism is a really important goal, and we'll be able to get there before too long.
I have one last question, and it's okay to be a little speculative in this forum, if you wouldn't mind. So the prevalence of autism has been increasing per number of live births.
Yeah.
And in parallel with that, as a society, we've been eating more and more processed foods, and we've gotten more and more away from whole foods, which of course has a profound impact on the gut microbiome. Any thoughts about that, why this prevalence is increasing, how that may relate to diet overall? And then back to some words of wisdom of what parents could really try and work toward today while we're waiting for all this wonderful science to emerge.
Yeah, so it's absolutely true about autism rates increasing rapidly, and it's not just autism. It's obesity, it's diabetes, it's inflammatory bowel disease, it's multiple sclerosis. It's a whole lot of different conditions that are all linked to the immune system and linked to the microbiome. So the problem is that it's difficult to tell which of the many factors that are changing at the same time are primarily responsible for that. Is it ultra processed foods? Is it increased use of antibiotics? Is it increased use of C-sections? Is it decreased breastfeeding? All of these different things we know impact the microbiome in ways that can potentially increase that whole suite of inflammatory conditions. In terms of what you can do right now, the general guidelines about anti-inflammatory diet, especially Mediterranean diet, which has been shown to be effective for a number of things, including major depressive disorder, interestingly. General guidelines like that are very, very helpful, especially in terms of setting a dietary pattern, rather than hoping that there's one magic ingredient that will solve all of your problems. And especially being able to make diets around whole foods that contain all of the micronutrients that are stripped out during processing, and where only the ones we know about are resupplied, but there's literally millions of other compounds that some of which may be important and we don't know yet. I think we're right on the edge of a revolution in nutrition, where just like a century ago, all of the things that led to then prevalent horrifying diseases like rickets, like pellagra, like beriberi, and so on, the nutrients that were responsible for those diseases when they were in deficit were identified, and it improved the health of literally millions of people. I think the diseases that were unknown then but are prevalent now, including things like MS, autism, IBD, and so on, it seems very likely to me that in the same way, there's something that we're stripping out of our food supply at the moment, where if we can identify it and resupply it and feed our microbiomes the right way, it could have a huge impact and make those diseases as rare as like rickets or scurvy or pellagra now, where you're just never going to meet anyone who's been affected by those diseases.
Well, thank you for that very hopeful answer. We like to say in our practice that there are 39 trillion microbes in our body and 30 trillion human cells. and they've been around for millions of years, maybe even billions of years longer than us. We like to think that we're hosting them, but maybe in reality, they're hosting us. So we really need to take good care of them and help them take good care of us. So with that, thank you so much for joining us. Great reply to a lot of these questions. And we'll get back to everyone and answer that question about dairy. And for those in attendance who arrived late, this evening's presentation is being fully recorded. Everyone will receive an email tomorrow announcing our one remaining Q&A event in this series and how to sign up for it and how to view all 6 pre-recorded webinars and Q&A from our top experts in autism. If you don't want to wait for that email, Dr. Knight's webinar, Autism and the Human Microbiome, can be viewed on our website at www.potomac.psychiatry.com. Once you're on our site from any page, click on the resources link and click on videos, and you'll get to the webinar. Thank you, everyone, once again for participating, and have a good evening.